So today I'm going to be talking about our research that we do at IOCB Prague, and there's going to be a little bit of biology, a little bit of biochemistry, a little bit of chemistry. So I'll try my best to make it understandable and digestible. We'll see how it goes. But first of all, I just wanted to spend one slide with a little bit of a self-introduction. Okay, it will make more sense later in the talk. So I originally studied computer science here in Prague at Matfis, and I was planning to become a software engineer. But then, for complicated personal reasons, I ended up in Japan. And because I needed a job, I actually eventually found a job in a lab that was doing molecular biology. So I became molecular biologist, and that's where I started learning about experimental techniques like mass spectrometry for measuring molecules, and eventually ended up doing my PhD there. Okay. And after that, because I learned something about how we measure and detect molecules, I found that kind of exciting. I decided to do a postdoc in the US, in Boston, at the Whitehead Institute, where I was working in a lab that was studying the chemodiversity of plants. And plants produce a lot of different chemicals, so it sort of made sense. I learned a lot more about how these chemicals are produced. And about 6 years ago, I started my own lab here at IOCB Prague, where I am trying to sort of combine these different fields and approaches that I encountered, and that's what you will see in the talk. Okay, so I want to start by saying that plants are amazing, right? If you type nature in Google Images, you get something like this. And you may notice there is a lot of green in these pictures, right? Why is there a lot of green? Because actually plants constitute the vast majority of biomass on this planet. Not everyone realizes that, okay? So that's first amazing thing about plants. Now, the second amazing thing is that plants cannot walk like we do, right? Obviously, cannot walk. I'm writing generally cannot walk. There are some exceptions. Like, does anybody know any exceptions? Yes, right. The triffids. I don't know if you have read this book, but it's a very good book, very classic kind of post-apocalyptic science fiction story about plants that learn how to walk and they start terrorizing humans. There's also a movie based on the book. The movie is not very good, but the book is very good. I highly recommend. Okay, but we generally don't encounter plants like this in our life, so the point still stands, plants cannot walk. And why am I saying this? Because that means plants rely entirely on chemistry for their defense and for their adaptation to changes in the environment. And over 400 millions of years of evolution, plants had the opportunity to explore a huge diversity of biosynthetic pathways, enzymatic systems, to produce a huge diversity of different molecular scaffolds, which give them particular advantage in their particular ecological niche. And because these molecules somehow tend to be bioactive, they tend to hit some protein or some receptor and so on, sometimes they also turn out to be useful for us humans as medicines, for example. Here's a couple of well-known examples. I just want to highlight artemisinin, which is a first-line drug for malaria. And the discovery of artemisinin was actually rewarded with a Nobel Prize in 2015. And the market size of artemisinin therapies is in the order of hundreds of millions of dollars per year. Or another example, paclitaxel, which is a chemotherapy drug, and the market size of paclitaxel therapies is in the order of billions of dollars per year. But if you take a look at the chemical structure of paclitaxel, you may realize that producing molecules like this is actually quite difficult. They cannot be easily produced with organic synthesis. And so oftentimes these molecules are still being harvested from the original plants, which obviously leads to problems with production and with over-harvesting and so on. So nowadays in the field there is a trend how to overcome these limitations. And instead of relying on organic synthesis, we study these medicinal plants that make these amazing molecules, and we try to use modern scientific approaches like transcriptomics, metabolomics, to elucidate the biosynthetic pathways of these molecules. So we want to learn how the plants produce these chemical scaffolds. Yeah, and when we know these biosynthetic enzymes, then we can engineer them into laboratory microorganisms such as yeast. And these yeasts, they're easy to grow. We can grow a ton of them, and then they start generating the chemistry for us, so we can produce sort of nature-inspired drugs or their derivatives without doing any organic synthesis at all. So it sounds wonderful. It's like a very green approach to chemistry and to exploring and exploiting the richness of the chemistry in nature. And there are some success stories out there, such as the artemisinin that I mentioned, or opioids, colchicine, which is another anti-cancer drug, vinblastine, and the Taxol, the paclitaxel. So there are some examples like this. Nevertheless, this has not really become a mainstream approach yet, because there is a number of bottlenecks. And one of the bottlenecks is that it's actually very difficult to figure out the biosynthetic pathways of these molecules in the plants. Because plant genomes tend to be huge, and the genes that interact in a particular pathway are not organized into clusters like they are in bacteria. So identifying these genes is actually a major challenge. So my lab is trying to look into this diversity and biosynthesis of these molecules, and we're trying to answer some very fundamental questions. So how do we measure and interpret the complex metabolic diversity in plants? It's like the angle of small molecules. How do we elucidate the biosynthetic enzymes, pathways, or networks? So that's the angle of the genes and enzymes. And most importantly, how can we do this at scale using computational approaches? Because traditionally, these things have been done in plant labs, kind of like one PhD student, one gene. And after 3 years of PhD or 4 years of PhD, they publish a paper, they characterize one gene. Yeah? But the number of genes that we have from sequencing experiments is now growing exponentially. So this is really not the pace that we can solve this. We need more high-throughput computational approaches. So I would like to start by talking about this Tropicana project that we started last year. And this is basically a project looking at this chemical diversity. So we go to botanical gardens, we sample different plant species, we analyze them on mass spectrometry. I will talk about that a bit more. And then we use computational tools that we also develop, like Enzymine and DREAMS, to find some new, unique chemicals in these plant species. So if we want to do that, obviously we need first the plants themselves. So we are collaborating with the Botanical Garden in Prague and also at the Czech University of Life Sciences. And through them, we have access to about 15,000 different species. Recently, we started a collaboration with the Kew Gardens in London, which is the largest botanical garden in the world. And it's actually really well equipped. They also have labs, they have lyophilizers, they have mass specs, they have PhD students working there. So this is a great place. And we also have collaborators in other countries, like in Switzerland. So we have potentially access to a lot of plant species. Obviously, we cannot sample them all, so we need to somehow prioritize. For that, we developed this workflow where we start with the Angiosperms Tree of Life that was actually published by the Kew Gardens. And we search public databases like Wikidata or publication databases like Scopus to annotate this tree with the number of natural products, molecules that have been detected or reported in these species, and number of papers that we have about these species. And we get sort of an annotated version of this tree, which looks like this in practice. So we have the tree of life, and then we have the papers and the natural products. And this is an example of a well-characterized lineage where we have a lot of papers published and a lot of natural products reported. But there are some lineages like this where there's almost nothing being reported. And these are interesting for us because we think there is a higher chance to discover some new chemistry. Why this is important? Because if you go to a botanical garden and you ask the curators in the botanical garden, please show me some interesting plants, they will show you something like this. Because for them, interesting means something that's well studied. For us, it means something that hasn't been studied. So that's why we have to do this. Okay, so we collect samples from botanical gardens, then we analyze them using mass spectrometry, and we get a lot of data. This data is then linked to all the metadata that we can collect. So that means the metadata that we receive from the botanical garden, but also geolocation data, photos that we take of all the angles of the plant. And this is uploaded into a knowledge graph that's organized by this network called Digital Botanical Garden Initiative that was started by these two guys in Switzerland. And they actually have a dedicated cell phone app. So when you go to the botanical garden, you can take pictures of the plants. It automatically stores the geolocation data and everything. You take a picture of the label, and everything is uploaded to the servers. So in one day, you can sample a couple of 100 samples this way. Otherwise, if you have to take notes, it would take much more time. So this is very efficient, and in this way we currently have about 700 plant species collected, and we're expanding this collection gradually. Okay. So now all this data is analyzed by mass spectrometry, as I mentioned, and how many people here have experience with mass spectrometry? Okay, some. Good. So for the others, very quick tutorial. This is a mass spectrometer, and this is another machine that's called a liquid chromatograph. The purpose of the chromatograph is to separate molecules. The purpose of the mass spectrometer is to measure the molecules. We start with, for example, an extract from a plant. It contains thousands of different molecules. This goes into an autosampler, and then it's injected into a flow of liquid that's pumped by these pumps under high pressure with these solvents through a separation column that's in this thermostat, an oven. And in this column, the molecules get separated by their chemical properties. So when they come out, they come out in batches based on their polarity, for example. And then they go into this ion source where we have high temperature and high voltage. And here everything is evaporated and turned into ions. So we have charged particles that go inside a mass spec where we have high vacuum, and we have some ion optics, some electromagnetic fields, so we can manipulate with these ions using the electromagnetic field. And in the end, we have some detector that measures their molecular mass. So all in all, it's basically a very fancy and very precise scale that measures the mass of molecules. And this mass then tells us something about what the molecules are, and we can also measure how much of the molecules we detect. This is how the data from mass spectrometry looks like. So we have the dimension from the chromatography, from the separation column. We have the dimension from the mass, so this is the molecular mass of the molecules, and we have the dimension of the signal intensity. So as you can see, this is quite messy and quite difficult to navigate. Right? So this raw data is actually not very practical for biologists or biochemists. What we need instead is some organized way of looking at the data, like a table of molecules that we detect, or some organization into networks based on some structural properties that we can predict, and so on. So to turn this raw data into some interpretable data, we use a software platform that's called MZMine. And this is a software tool that I have personally been working on for almost 20 years. It was started in 2004 by a research group in Finland, and then I took over the development of this project around 2006 when I was in Japan. And as you can see by the number of lines of code, we have been developing this with more and more people joining the project from various countries over many years, and it has actually been accelerating. So this has gradually become a kind of very, very versatile tool to process all kinds of mass spectrometry data. This is a screenshot of the latest version. So you can see it has lots of controls, lots of features, and we published this in 2023 in this Nature Biotechnology paper, and the title was Integrative Analysis of Multimodal Mass Spectrometry Data. What that means is that you can use any kind of mass spectrometry data. There are many types of mass spectrometers, and you can integrate them together. So you can actually look at the same molecules with different sort of points of view. And MZMine is currently the only software that can do this. Because the data processing then becomes very complex, we also have tools like MZ Wizard, where you can just configure what kind of experiments you're doing on the mass spectrometer, and this sets up the data processing workflow for you, so it's very easy to use. Well, easy to use, but nevertheless, it's still quite complicated. So we also published this paper in Nature Protocols, which summarizes all the ways how you can process the data using MZMine based on the technology that you're using to detect the molecules. Okay. So this gets quite complex. And I think at the time we sort of realized that this whole project has become so big that it's impossible to maintain it with PhD students and volunteers in academia. So it was around 2004 that we decided to start a company. It's called MZIO. It was started in Germany together with the main developers of this project. And now the development of MZMine has been fully transferred to the company. The software is still available for free for academic users, but now we also have an enterprise version of MZMine that's offered to industry and biotech and pharma customers. Nevertheless, we like to stay connected to the academic community, so we organize these workshops at IOCB Prague every year, where we show people and teach people how to use the latest features in the software, and anybody can join and learn something. And we don't do this only in Prague, but we actually do it all around the world at various events, workshops, and conferences. And now that we have the company, we actually try to be present at all the major conferences in the field to have our demonstration booths so that we can spread the news about the software. So I think this is quite a nice story how an academic open source software can mature and actually be turned into something that's really professional and enterprise level. This is the last example from a conference of Metabolomics 2025 that was in Prague last year. And we organized the workshop on MZMine together with members of my lab and the company MZIO. As you can see, it was pretty packed. So now the question is, okay, we are getting pretty good at processing and understanding mass spectrometry data. So what's remaining? What are the research problems? And when I started working on metabolomics in 2006, people told me that the biggest problem really is annotating the metabolites. We can see a lot of molecules in the mass spec data, but we don't know what these molecules are. Now, fast forward 20 years, and in 2026, if you ask the experts, they tell you exactly the same thing. So the biggest bottleneck is annotating the metabolites. Okay. Why is that? Why we haven't really solved this problem in 20 years? Well, I think it's because we have very small amount of actually reliable reference data. So typically the annotation of the molecules is done by comparing their spectra between your experimental spectra and some reference spectra that you have in the library. And here is a breakdown of all the chemical structures of known natural products. And these are chemical structures that were reported in humans, so human metabolites. And these are the chemical structures that you can find in publicly available spectral libraries. So you can see this is a very small coverage. And this hasn't really been improving over the last 20 years. Okay, so this is a problem. What can we do about it? Well, obviously, we can build bigger and better spectral libraries, so we have more reference data. Or we can try using unannotated data for some machine learning magic, so we can learn from the unannotated data. And in my lab, we're trying to do both. So I'm first going to tell you something about building larger spectral libraries. For this, we developed an experimental workflow using a pipetting robot that pipettes 10 compounds in a single well in 384 well plates. And we use large chemical libraries that we mostly received as donations. And then we inject these mixes of compounds into the flow of the chromatograph. And we see the compounds for about 90 seconds, during which we collect all the spectra that we can that are possible on these compounds. And then this is followed by a data processing workflow, of course implemented in MZMine, where we take all this raw data, we do some quality checks, we isolate the spectral trees of the individual compounds, and then we export it in a kind of standardized library format. This is an example how the data looks like. We take the molecules, for example, this molecule here, we fragment them by colliding them with a gas. We get spectra of all these fragments, then we fragment them again, we get even smaller fragments, we collect their spectra and so on. So this is actually a very rich representation of the data of those molecules. This was published a few months ago in Nature Methods, where we reported the library of 30,000 molecules, about 2 million spectra, so we have almost 100 spectra per molecule. And just this paper has basically doubled the amount of reference data that's available for the whole community that was produced by the last 20 years, let's say. Nevertheless, we are not stopping there, and we obtain more compounds, so we're continuing this analysis. So at this point, we have about 70,000 compounds already analyzed, and 150,000 more waiting in our freezer to be analyzed. And if any of you has an uncle who has a chemical company, would like to donate some chemical compounds, then please let me know. We are welcome to add more compounds to this collection. And all the data is published online for free with no restrictions. Okay, now the second approach that I mentioned is based on using unannotated data. For this, we use a repository that's called Massive, that's run at the UCSD in San Diego, and that currently contains about 500 terabytes of raw mass spectrometry data. And the only thing that we know about the data is where it comes from. So it could be a food sample, or it could be a human sample, or a plant sample. But you don't have any annotation about the molecules or about the spectra. You just know something about the sample. So for this, we developed an approach that's based on machine learning, similar to the approach of large language models. So when you train large language models like ChatGPT, typically it's done using something called masked language modeling. So you take a lot of text, you hide certain words in the text, and then you ask the model to predict the missing words. In order to do that, the model has to learn something about the connections between the words, about the context, about the structure of the language, and so on. So if you keep doing this using large amount of text, you get a model that sort of understands the language. Yeah? So we took the same approach, and we applied it to this huge amount of mass spectrometry data. And what we're doing is we are hiding certain peaks in the spectra, and we are asking the model to predict these missing peaks. Yeah? In order to do that, the model has to understand something about the chemistry, like how these spectra are actually produced, how they relate to the chemicals that they represent. Yeah, so the model only sees the spectra, but it has to learn something about the language of the spectra in order to do this task. And because we have a lot of data, we can keep training this, and we get a model that sort of understands spectra. Of course, in reality, it's a little bit more complicated, because, for example, in mass spectrometry, we really care about the precision of the numbers. So when you measure a molecule, you really care about the second and third decimal of the mass measurement, which is a different situation from language. In language, you don't have this problem because you're dealing with just words. But in mass spectrometry, we really care about the numbers. So we had to do some technical tweaks to make sure that the precision of the numbers is not lost during the calculation by this neural network. Neural networks are chronically bad at calculating, so took a bit of engineering. But in the end, we have some model architecture that actually does the job and predicts the missing peaks. Yeah? But that's not really useful, like predicting missing peaks. Right? But we have something that sort of understands the spectra. What that means is now we can take some experimental spectrum, and we can use our model, we call DREAMS, to calculate a vector representation of the spectrum, so some called sort of embedding. And we can lay out these embeddings for different spectra in a 2D space. And the interesting thing that we observe here is that the spectra start to be organized by chemical structures. So for example, all the halogenated molecules are here. All the carbohydrates are here. But the model has never seen chemical structures. The model has only seen the spectra without any annotation, but naturally it started organizing them in a way that makes sense in the context of chemical structures. What this means for us is that we can use this embedding as a metric of molecular similarity. So for example, here we have two different spectra, and our model places them very close to each other in the embedding space, unlike these two spectra, which are very far away from each other. So it means our model thinks that these two spectra have something in common, even though they look completely different if you look at them. Okay? Well, when we see the chemical structures, it actually starts making sense, because these molecules, although they are different, they are composed of very similar building blocks. So the model somehow understood, despite the spectra being completely different, that these molecules that are hiding behind the spectra have something in common and should be close to each other. So we can now use this as a metric for chemical similarity of the molecules that we are measuring. And you can do this analysis, of course, in MZMine. So you can analyze some samples on mass spectrometry, load the data into MZMine, and then take any molecule that you see and start building networks of similarity with other molecules. And this helps with interpreting the data. What we can also do then is apply this in a large scale. So we built a very large molecular network using this DREAMS similarity from 200 million mass spectra from the massive repository. And this is kind of like a really big network. You could think of it like the internet of mass spectrometry of small molecules. So we have all these spectra here, and these are spectra coming from many different studies, from many different labs, many different experiments, and many different samples. And you can use this data set to form some research hypotheses. So for example, here we have some spectra, these are the nodes, connected with high similarity into this small network. And these spectra come from some plants, like a tree or fruits and vegetables. They come from human tissues, face, hand. And we have a disease sample, psoriasis is a skin disease. And we have this pesticide here and the pesticide mix. Yeah? So when you get this kind of connections, even though these data sets may have been collected by different labs, you could make a hypothesis that, okay, maybe the origin of this disease might have something to do with the use of this pesticide on the fruits and the plants, through which they go to the human tissues and could have something to do with the start of this disease. The cause of psoriasis is actually unknown. I'm not saying that's the case, I'm just saying you could make that kind of hypothesis. Another thing you can do is you can take the vector representations of all the spectra that you collect from one sample, and you can average them. If you average all these vectors, then you get one vector that represents the sample. It's sort of combined representation of all the molecules that you detected in that sample. And you can do this for many samples. So now, here we're not looking at spectra, but we're looking at samples. Each of them contain thousands of spectra. And then again, you see that these are food samples, so many different types of foods. Again, you see they start being organized. So we have all the beverages in one place, all the animal foods in one place, plant foods in another place. So again, the representation of the sample somehow reflects the chemistry that's underlying. This is kind of a very high-level view of the samples. So we published this analysis in this paper last year in Nature Biotechnology, and the guys Roman and Anton here are now developing a sort of web interface that people who are doing mass spectrometry can easily use, upload their data, and do this kind of computational analysis by themselves without needing any coding. What else can we do with this kind of model? Because I said we have now a sort of foundational model that understands spectra, but how can it be useful besides calculating similarity? One application that I'm particularly excited about is for finding new chemistries in plants. So we start with many plant samples, like in the Tropicana project, and we're asking what are the most unique or most novel molecules that we can find. And we can do it by, again, doing mass spectrometry, looking at these DREAMS representations of all the spectra, and looking at the outliers, and we are comparing the representation against some reference space. What is this reference space? That depends how you define What do you want? What is novelty for you? It could be the space of all the annotated spectra, or it could be space of all the spectra that have been previously detected in plants, or it could be space of all the spectra of molecules that we already know chemical structures of. So there are different approaches to this, but basically in any case we are looking at these outliers, and then we know that these molecules will be something that's exciting, right? It will be something that's worth isolating, purifying, and subjecting to more deeper structural analysis, because these things take time. This can be 2 years of work, so we need to know which molecules are exciting enough that we actually want to invest this time. This is just one example. This is a spectrum from this pepper plant, and this is the closest spectrum from our huge library of millions of spectra, which already corresponds to a pretty weird molecule, but is still quite far away from this experimental spectrum. Now, I don't know yet what this molecule is. Maybe in a few months I can tell you. Now, another thing that we hope we can do is we can hopefully get to real chemical structures directly from the spectra. This is something that many labs in the world are trying to work on, and nobody has really cracked this problem. Think about AlphaFold. Probably you heard about AlphaFold. It's a very sophisticated model that can predict protein structures from their sequences. But it doesn't only give you the protein structure, it also gives you some sort of confidence metric. So you know which parts of the structure you are confident about and which part of the structure you're less confident about. So we hope we can use this DREAMS representation to take a mass spectrum as an input and generate de novo chemical structures, which are complex graphs, so this is actually quite a difficult task, with some confidence metric. Because we can be pretty sure that the information that is contained in the spectra is not going to be enough to always get the full structure right. But if we can at least know that some parts of the structure we know for sure, and some parts of the structure we are less confident about, this would already be a huge progress. We have been working on this, and at this point we have some version of this DREAMS small model that generates chemical structures, and it is beating all the other competing methods in an unbiased benchmark. This benchmark actually didn't exist, so we all actually had to build this benchmark ourselves together with colleagues from around the world in order to make sure that we are really making something that is performing well. Yeah. So we teamed up with many different colleagues. This was a collaboration of many institutions around the world. And we built this benchmark, which contains a data set. So we have some reference spectra and reference molecules. It contains very carefully designed splits of the data set. So we define which part of the data you should use for training your models and which part of the data you should use for evaluating the performance. And we took a lot of care to avoid any kind of data leakage from the training to the testing, which is a big problem typically in these public data sets. And then we define some tasks that the models can solve. And then we have a leaderboard where we show which models are performing the best on these tasks. The best performing model for the chemical structure prediction is this DIFMS model coming from MIT. That's just because this DREAMS small model has not been published yet. So I hope this will change in a few months. So it's very easy to implement a new model for predicting chemical structures and just plug it into this benchmark, and you will see how well you are doing. Okay, so this was the first part of my talk that was focused on how we detect molecules, how we map this chemodiversity. Now I want to switch gears a little bit and talk about the biosynthesis. So how do the plants do it that they generate this amazing diversity? For this, we know that plants contain a lot of proteins, a lot of enzymes. Each enzyme has a sequence, so this is written in the genome. So we have a sequence like this. That's very easy to measure. We just sequence the plant and we get all these sequences. But to annotate the sequences with function, that's very difficult. So ideally what we want to have is some sort of model that takes the sequence as an input and then tells us this sequence codes for an enzyme that's a sesquiterpene synthase that takes exactly this substrate and produces exactly this product. And if you have some experience in biology, you could say that tools like this already exist and have been around since the 1990s. Maybe some idea which tool? Like BLAST, for example. BLAST is a sequence search tool that's very efficient. You can search large databases of sequences. So you just take the input sequence, you blast it, and you get some annotation. The problem is that it only works if your query sequence is very close to your training data, to your annotated sequences. And moreover, it only does like pattern matching, string matching. It doesn't really give you any interpretation or any understanding. Nowadays, we have seen that there has been like a huge revolution of deep learning methods for biology. We can now predict structures of proteins, including nucleic acids and ligands. We can generate new protein sequences that will fold into particular shapes, like letters of alphabet, or we can design binders based on some motifs. So we have seen this like huge revolution, but this revolution hasn't really been much reflected in the space of enzymes. Enzymes are a bit more complicated than other proteins, because to make a functional enzyme, you not only have to have the right sequence that will fold into the right shape, but it has to have the right active site that's going to bind the right substrate, and then it's going to have the right dynamics to catalyze a chemical reaction. So it's sort of a very complicated area. And we see some people working on this. So there are some sort of state-of-the-art methods for predicting enzyme functions. And these state-of-the-art methods are typically based on contrastive learning. So they try to deduce some annotation from the protein sequence using contrastive learning and generate something that's called enzyme commission numbers, which is a hierarchical annotation system for enzymes, where these 4 hierarchies come from the most general. So this could be something like oxidoreductase, to the most specific, which this could be the actual chemical reaction that the enzyme is catalyzing. And there's a number of models that have been published. The most famous one is this clean model, probably. That was published in Science, and they show how well it works for the different levels of the EC hierarchy. But if you take a step back and you think a little bit about what these models are actually doing, this is the space of all protein sequences, and then some of them, the red ones, are annotated, so we know what they do. Now, let's say we want to annotate this blue protein, that's our query. So what we could do in 1990 using BLAST is we could find the closest matching training sequence. What we can do now with these fancy contrastive learning methods is we can take a more closer look at the neighborhood of the query sequence and make a more informed decision which one of these is actually closest to the query. So it works better than BLAST. There's no question about it. It works better than BLAST. But still, it has fundamental limitations. Like, you will never be able to discover completely new reactions, because they don't even have EC numbers. Nobody has assigned EC numbers to unknown reactions. It does not generalize further away from the training data, because you don't have anything to learn from. And again, it doesn't give you any kind of interpretation of the biochemistry. It just gives you some probability number. So in my lab, we're trying to approach this problem in a slightly different way. And instead of trying to solve all enzymes, which I think is currently impossible, we're trying to look at a specific family of enzymes and learn from everything that people have published about the biochemistry of these enzymes, so we can input this into the machine learning process. So the class of enzymes that we are looking at are called terpene syntheses, and these are enzymes that take these linear pyrophosphate substrates and they cyclize them into scaffolds. They are called terpenes. And then these scaffolds can be further decorated by many types of additional enzymes to produce a variety of compounds called terpenoids. There's over 80,000 of known terpenoids. It's actually the biggest class of natural products that is produced by almost all species on this planet. And that includes paclitaxel and artemisinin that I mentioned in the beginning of my talk. So we are looking at these enzymes that take the linear precursors and produce these scaffolds. And we would like to predict their functions using machine learning. So obviously, we need to start with data. So how many terpene synthase reactions have been characterized by the whole biology community to date? Well, that's easy. You just go into a database of terpene synthesis and you look at how many entries are there. Except such database didn't exist, so we had to build it ourselves. So we had a number of students who started working 6 years ago on this Google sheet and typing information that they found in papers and basically curating all this knowledge that we have about terpene synthesis. And because this has taken a lot of effort and time, now we have 1,300 characterized enzymes that catalyze 2,600 reactions. And we actually turned this into a web-based database that's called MarsDB, and we really collect all the information about terpene synthesis, not just the substrate and the product, but also the reaction mechanisms, so all the intermediates, active site residues, everything that is known. So yeah, like I said, we have about 1,300 enzymes, most of them coming from plants and fungi and bacteria. This was just recently published in a paper in BMC Bioinformatics. Okay, so now we have data. Again, 1,300 enzymes is really not the scale of data that we could apply some deep learning methods. But what can help us is this language model approach. But this time, we're not going to be building language models for spectra, but for protein sequences. So again, similarly to ChatGPT, we start with a huge number of actual biological protein sequences. We hide certain amino acids, and we ask the model to predict the missing amino acids. In order to do that, the model has to understand something about the structure of the protein, about the interactions of the residues, and so on. And because we do this for a long time, we get a model that sort of understands proteins. Now, the good news is we don't actually have to do this because there is a number of these protein language models that have been already trained and designed by experts, so we can just use one of these. And now it seems like the whole task is actually quite easy. We take the input protein sequence, we apply the protein language model, we get some sort of vector representation, and then we put a classifier or a regressor on that vector representation, and that gives us all the answers that we want, right, about the function. Except it doesn't really work. I mean, it works to some extent, but it doesn't really work well enough to be useful in practice. And there are actually papers that show that you really have to consider the structure of the protein, not only the sequence, because in terpene synthesis, the outcome of the reaction is really sensitive to changes in the structure. Well, we know that terpene syntheses are composed of 3 structural domains. So this is the structure representation, and we have the alpha, beta, and gamma domains. So this is kind of a textbook knowledge about terpene synthesis. So we decided to look at this more in detail, thanks to the fact that we have a database of all the terpene syntheses that are known. So we started with our database, we predicted all the protein structures using AlphaFold, and then we used the templates of the alpha, beta, and gamma domains to identify and cut out all the individual domains in all these proteins. So now we have all the individual domains, and we do a pairwise structural alignment, and we do clustering on these results. And not surprisingly, we see a cluster of the alpha domains, cluster of the beta domains, and the gamma domains. And somewhat surprisingly, we found 2 additional clusters. So we started calling them delta and epsilon. And these have never been reported in the literature. Probably people knew they exist, but nobody just mentioned them. And I can show you that they really are different. So this is a typical diterpene synthase with the alpha, beta, and gamma domain. And this is a typical triterpene synthase with the delta and epsilon domain. And they really look quite different. Okay. Now we can do this analysis with a bit more fine resolution. And what then comes out is that we can actually recognize some specific subtypes of these structural domains. So we don't only have one alpha, but we now have alpha 1a, alpha 2a, alpha 2b, and so on. We have different subtypes. And we recognize about 20 different configurations that the terpene synthesis can have. Some of them are more common, some of them are quite rare, and they differ by the distribution in the different kingdoms of life, and they also differ by the biochemistry that they catalyze. But this was quite an interesting analysis, because with this, we made a sort of a map of all the biochemistry of terpene synthesis from a structural perspective. So now we know something about how the structure relates to function and what kind of structural motifs we can recognize, and we can use it in the machine learning predictions. So this is how the prediction pipeline looks like. We start with the input sequence. We predict the structure of the protein using AlphaFold, and then we detect all these individual domain subtypes that we now recognize, and we encode this structural information into a vector. At the same time, we use the protein language model, and we encode the sequence information into the vector. So we now have two vector representations, and from these we make predictions, whether that given protein is a terpene synthase or not, and if yes, then what is the substrate. And these classifiers here are trained on our training data on the 1300 examples. And because these are pretty simple models, their 1300 examples is actually enough to get good performance. Well, when I'm talking about performance, we have to compare ourselves to some existing methods, some baselines. There is actually a method called Terzyme that's using hidden Markov models that's trying to solve exactly the same problem. So detecting terpene synthesis and predicting their substrates. There is a structural approach called FoldSeq that's used to compare protein structures. So we can use that as a baseline for the structural comparison. And there is this clean method that I mentioned that predicts the EC numbers. So we can use that as a baseline as well. And now, if we compare ourselves on the problem of terpene synthase detection, we are doing better than all these baseline methods. And the same for substrate prediction. So this is very nice. We have something that performs the best. But of course, you should never really trust these plots if you see them in publications, because it's very easy to make the evaluation look like you are doing the best, right? So I think that's, of course, applies here as well. You should not trust me. But what really matters is, can we use this to do some new biology? So since we have the terpene synthase detection that shows almost perfect performance here, we decided to go back to sequence databases. So we started with UniProt, which contains 200 million sequences. And we selected those sequences where our model says that they are terpene synthases, but every other bioinformatic tool says nothing. So basically, we call this the dark matter of sequence space. We use all the bioinformatic tools that are combined in Interpro, which is an ensemble of bioinformatic information annotation methods. And we selected those sequences where Interpro returns 0 results, not even a domain of unknown function. So we took 20 such sequences, and then we produced the genes. We expressed them in a microorganism in yeast. And we start looking, using mass spectrometry, in this case GCMS, whether these enzymes actually start producing any terpenes. And indeed, we found sequences that, despite having no annotations, start producing terpene signals on the GCMS. And this particular one is coming from a virus. That was very exciting, because viruses are generally not recognized for having these metabolic enzymes that actually generate some small metabolites. This is the first time anybody has ever seen something like this. So we were very excited to have discovered the first functional viral terpene synthase. And then a few weeks later, there was a paper coming out that actually described this. So now we know we discovered the second functional viral terpene synthase, but it's still quite cool. And we also discovered some functional terpene synthases from Archaea, which is another kingdom where no terpene synthase has been discovered to date. So we have some functional examples now, and we also know something about the molecules they produce. Okay, so this is quite exciting. We have this in a form of a preprint. We're trying to get this published now. But you may remember I was talking about functional annotation of proteins, but so far until now I was only talking about the substrate and not the product. So what can we do for predicting the product of the reaction? in the case of terpene synthesis, this is the substrate, this is the product. You can see this is going to be very difficult to predict, right? Because there's so many transformations that happen to change this molecular graph into this molecular graph. And predicting a transformation like this using only 1,300 of known examples, I think that's pretty much impossible. What we can do instead is we can look at the reaction mechanism of the enzymes. So the reaction doesn't happen in one step, but from the substrate, we have certain transformations that make biochemical sense, and then they are followed by other transformations, and in the end, based on the probabilities of these, which are dictated by the enzyme, we get some distribution of possible products. And typical terpene synthase has more than one product, so this is actually a realistic example. And because we have all this data about these mechanisms and the distributions of the products in the MarsDB database, we can now look at the enzyme space from the perspective of what we call a reaction forest. So these are not reaction trees of individual enzymes, but this is the reaction forest of all the trees combined. Starting from one common substrate, we have all the enzymes here that lead to different products that are colored in green. And if we talk about one particular terpene synthase, we can think of it as one particular path through this tree. And another terpene synthase takes a different path and leads to different products. So now this whole problem of product prediction hopefully becomes more tractable in the sense that in each step, given some enzyme that is surrounding the reaction, and given this intermediate structure that's currently in the active site of the enzyme, what we need to predict is just what happens next. What is the next possible step, next possible transformation? And the good news here is that there may be only few examples of what makes biochemical sense. So instead of predicting the whole molecular graph, which is very difficult, we're just predicting a couple of options of what happens next, or the probabilities of these options. Now, this is something that we are actively working on now, so I don't really have any results that I could show you, but stay tuned, and maybe if I come again in like 2 years, I can show you how well it worked. So with this, I would like to thank my amazing lab, and especially our collaborator, Josef Šivec from CIRC here at ČVUT, who has been really instrumental in designing with us these machine learning workflows. And I'll be happy to answer any questions. Thank you.
Thank you, Tomáš, for an amazing talk. In my head, it was like 5 different talks. Questions?
Maybe you use the microphone so they can hear it. Microphone, yeah.
You work with botanical gardens, but just because you want to know what are you studying, theoretically you could go to the collect some grass in front of the institute and study the compounds from there.
We do that as well, but the advantage of botanical gardens is that they have curated collections, so they know already some stuff about the plants that they have, like what species it is, although it's often wrong, but at least some information. And the second advantage is that if you go to the same botanical garden 2 years later, there is a high chance that you will find the same plant in the same spot still being there, which is not necessarily true for the plants in the wild.
I have one question. If you have like some species in the 2 different botanic garden, how they are different actually?
they will be different. They will be different not only if they're in two botanical gardens, but if you collect data from the same plant in the morning and in the evening, it's going to be different. If you collect it in the spring and in the autumn, it's going to be different. So obviously these are living systems and they are dynamic. Still, I think this is not really a problem for us because the changes that we see in the plants are more like quantitative changes. So you see different levels of different chemicals depending on what situation the plant is and how old it is and if it's a sunshine or if it's dry and so on. But there are not so much qualitative differences, in the sense that the plant would be producing completely different chemicals suddenly based on different situations. So the differences between the different species are still kind of much bigger than the differences in one species in different situations. So I think in terms of like discovering new chemicals, it doesn't really matter that much, like what is the particular ecological situation of that plant that we are studying. It can matter in case where we want to, for example, isolate the pure compound and we want to maximize the yield of what we're getting from that plant, then it's worth actually studying, under what conditions the plant produces the most of this particle chemical. But yeah, those are the quantitative differences.
If you have the phylogenetic tree of all of the plants and what we have now and all the spectra what you have got, are there kind of blank spots on it? Like some part of the phylogenetic tree which you have underrepresented and something which is overrepresented?
So the phylogenetic tree of life that I showed, it actually includes only about 50% of known plant species. And the Kew Gardens, they're preparing now to publish an updated version of this tree that is going to contain like 99% of known species. So this is also the taxonomy of plants is a very developing field, right? So that's one blind spot. We actually don't see many of the species on the tree, but that's hopefully getting better. There are some species that we simply don't have access to because they only grow in some remote place in Brazil, and somebody characterized them in 1960s, and then since then nobody else has ever seen them. So there are many examples like this. Right? This is if you take for example, 100,000 species that we can potentially collect from botanical gardens, they are sort of nicely distributed all around the tree. So yes, there will be some lineages that we simply cannot get access to, but in terms of sampling the diversity, I think it's okay. I think we can get a good representation of all the different genera and families and so on.
How does the preparation of a sample for mass spec work, and can you be sure that you can see all the molecules that are in the plant in your spectra?
We will never see all the molecules, that's for sure, because the chemistry is so diverse that if you want to cover all the different types of chemicals, you would have to really do many different types of extractions and many different types of analyses and so on. But we have sort of the common targets. These are small molecules that are, let's say, between 100 Dalton to 1000 Dalton, relatively hydrophobic, which is what most of these natural products are. And we extract them with organic solvents. And typically, if you use something like ethanol, you get most of these molecules out pretty well. I mean, there will be some that you will miss. That's life.
Maybe I have a related question about samples, about their treatment. Did you think about, for example, embedding your molecules into deuterated water? Then all protons are exchanged for deuterium. You have exactly the same molecule, but you can figure out better. For example, you can probably assign the molecule better because you have another information for the same molecule, or the protein that can be cut it into the pieces, and then you can read it or assign it probably with higher precision.
Yeah, I mean, this is an approach that people typically use for proteins. Like when you study protein structures, you do this hydrogen deuterium exchange to expose which residues of the protein are close to the surface and so on. It's not an approach that would be like widely applied to small molecules, but I don't think it's impossible. I think it should work, and it at least tells you something about how many hydrogens you have at what parts of the molecule. Yeah. I don't think there are any existing computational methods that could take such data and make some useful predictions regarding the structures. Right? That's something we might want to look into. But it's a nice idea. Yeah.
Okay. If there are no more questions to Tomáš, we all thank you a lot for a really inspiring and wonderful talk.
Thank you.